Thank you. Well, I want to thank everyone for joining us on this virtual seminar. My name is Jun Li. I'm a professor in human genetics and computational medicine and bioinformatics. I'm also one of the co-lead in the single-cell spatial analysis program. So this is one of our monthly seminars. In coming weeks, please keep your eyes open for announcements about other talks, including those presented by faculty candidates for the program. Today, it's my pleasure to welcome Dr. Rui Chen from Baylor College of Medicine to present a talk titled Single-Cell Spatial Map of the Mouse Retina. Chen, let me just cover his background. Ray received a bachelor's degree from the Tsinghua University and a Ph.D. from Baylor, followed by postdoc and faculty position at Baylor, where he's currently a professor in the Department of Molecular and Human Genetics. He studies the genetic basis of complex human diseases. In recent years, leveraging single-cell technology, he's currently the director of a Center of Single-Cell Biology and also the director of a single-cell genomics core. He's been active in national initiatives, including 3 projects funded by the Chan-Zuckerberg Initiative. He's a PI in the Retinal Network, the Pediatric Single-Cell Atlas of the Human Eye, and the third one is the Ancestry Single-Cell Atlas of the Human Eye. His work has been published in over 200 peer-reviewed publications, and we came to know his work more recently as an early adopter of the commercial offering of the Murfish technology. I think that's one component of his talk today. Ray, I want to thank you for spending time with us. Please take it away.
Thanks, Jim, for the invitation. It's really my pleasure to present here. I'm so impressed by the number of people actually dial in the Zoom, and given it's holiday today. And also 2 years in the Zoom, like we all have, I'm pretty much, have a lot of Zoom fatigue. So today I would like to share with you our recent effort using the Murfish technology to construct the spatial single-cell map for the mouse retina. And I will say we have encountered many challenges and then learned a lot. So hopefully our experience will be useful for your upcoming projects. So let me share my screen first. Can you see the screen okay?
Yes. Yeah.
Okay. So so our lab is interested in study, as Jim mentioned, the visual system, particularly in the retina. So the reason we are quite interested in the system is, of course, one of the reasons is that retina is the main target of irreversible blindness diseases. So the retina is at the back of the eye. That is the neuronal tissues that convert the light signal coming from the front of the eye to electric signal that's sent to the brain. So the light is captured at the very back of our eye and by the cells called photoreceptor cells. There are 2 types of major photoreceptor cells, cones and rods. The cone capture the basic color vision, and then the rods sense the light intensity. So the signal, the light captured by these photoreceptor cells convert to electric signals and then transmitted to internuclear layer interneurons, such as bipolar cells, and then it connect to and then sent to the RGCs, retinal ganglion cells, which send the signal to the brain, so we can see. So despite there's only essentially 5 major neuronal types in the retina, if you look at the based on the morphology and then the function, and this can be subdivided further into about 60 different types, neuron types in human. So those are the breakout. As you can see, the photoreceptor cells, the easiest ones. We have rod photoreceptor cells, and then 3 different cone receptor cells sense different light, 3 different light wavelengths. And then the most complex ones is amicron cells in terms of numbers, and then ganglion cells, which are one is in the inner nuclear layer, the other is in the RGC layer. So additional complex on that in terms of the identify individual cell types is also, although those 2 cell types, amicron cells and ganglion cells, have the most number of subtypes, they only made up a small fraction of the retinal cell types. For example, in the ganglion cells, just about estimate about 15 different types in our eye, but only account for 1% of the retinal cell types. So therefore, making identify individual cell types, it could be quite challenging. So this is actually further complicated. I mean, for reasons we don't really fully understand, but it seems that the number of cell types in the mouse is even actually exceeding that in human. So various of single cell RNA data has been study has been conducted, and then so this is a meta map our lab put together by collecting all the data published, major data set published, along with our own data. And then when you combine together, and those different colors represent the major cell types in the retina, and together there's more than over 100 different cell types in the mouse retina. Particularly, they have larger number of amicron cells and RGCs compared to human. Well, the photoreceptor cells actually identical. So, so given the the large amount of data people collected over the last 5 years using single cell RNA sequencing, it's clear that the transcriptome of individual cells reflect very well with the morphology and functional characterization. So therefore, the transcriptome is a good a method to identify individual cell types. So one problem with the single cell that this audience is probably very familiar with is that the single cell RNA-seq is you can identify individual cells on their transcriptional. However, one thing is missing is the spatial arrangement of that cell. Given it's a neuronal tissue, and actually for most of tissues, the spatial information of individual cells, in fact, is very critical. So therefore, we are thinking about how we can now take the knowledge we gain from the single cell RNA-seq transcriptome studies and to build a spatial map, so we can learn more about the function and mechanism of the retina. So we start looking at what technology are available to do that. There's based on the technology, we can roughly divide them into 2 different groups. One is the sequence-based, one is hybridization-based. So among the sequence group, basically you somehow isolate either a small group of cells or individual cells, and then you isolate their RNA, and then you make cDNA and you sequence, just like single cell RNA-seq, but you basically have maintain the spatial information. For example, the VCM and SNI-seq, DB-seq, and Elstrand. Particularly, the scope actually is developed by Michigan by Dr. Lee's group, which is really exciting technology. So the advantage for this method is, of course, they are unbiased, so they can sequence the whole transcriptome. And then, however, the one of the downside I will say is right now, for example, for the VCM or for many of those are not true single cell resolution, and then also because of the RT efficiency, the sensitivity of detecting transcript is somewhat compromised. In contrast, there's also hybridized-based method, such as the it's basically developed from in situ RNA hybridization, for example, Murfish and Sigfish, and also a set of other company which are basically popping out every month. So they have a lot in common, and because most of them are require only look at a panel. It is in theory possible, or actually in practice possible, to look at 10,000 transcripts. But most of the cases you're looking at anywhere from tens or hundreds of transcripts once you have the panel. So the main limitation here is you do only integrate the panel of genes you're designed for. However, on the other hand, they have, because they have in situ hybridization, the sensitivity of them can be achieved to 80% or higher, so they're very sensitive to picking up things. So today I will mainly share with as I mentioned, our experience using Merfish. So just a quick introduction of the Merfish technology. So Merfish stands for multiplex arrow robust fluorescent in situ hybridization. So basically with us, it's like for a set of genes you're interested, you design about 30 to 50 probes tile the transcript. And each probe from the same transcript, they have the same barcode, and they basically have 4-bit barcode on those probes. And then you do a series, once you hybridize the probe to the tissue, you can do a serial hybridization and basically read out which barcode the probe carries and which barcode you can detect in the tissue. And then based on the barcode, you can basically identify, or assign the individual the signal to the transcript, target transcript. So basically, there's a series hybridization, and then you can see from raw data, it's just basically pseudo-colored, spots, and then from the spots, which cycle you can detect spots, you can basically determine this transcript. So we're fortunate to in collaboration with Jeff Moffitt, who is one of the co-inventor of this technology, and then introduced to Vision, where they are trying to commercializing this technology, and they're making the technology called Merscope, and we are selected as one of the early access site, one of the 6. And here's what it looks like. It's basically totally open, and then you have 2 parts. One is basically the image parts. The sample is right here. And then you have the pump, fluid part, which you pumping different, probe in there. And as I said, there's a serious hybridization process and washing, and that's come up here. So it's quite simple. And right now they already commercialized the instrument, and they pretty much like this, but it make it more engineering, nicer or robust. I think the technology is pretty much the same. So just give you a brief view of the workflow we have with the Merfish experiment. I can show you. So basically, we start with the retina, and we start with the frozen OCT, flash frozen retina. In this case, the mouse, and then we basically make a frozen section at 10 micron in thickness, and then we fix the tissue and then hybridize the probe we pre-designed based on the meta-analysis we have done based on the RNA-seq data. So after hybridization, we also add a few cell boundary markers for reason that become obvious why we need that, and then that's basically our antibodies that against the cell surface marker, and then they conjugate with the probe so you can actually see them as well. And then lastly, we do a serial hybridization, decoding, and basically identify the transcript distribution, spatial distribution in this section. So this is what the raw data looks like. So this slide is a section of the retina, and the bright dots and the white dots are DAPI staining basically, and the highlight where the nuclei is at. As I mentioned, the retina is multi-layer structure. So you can see there are 3 layers in here, where this is the photoreceptor cell layer, where it's the back of the eye, and this is the inner nuclear layer, and this is the RGC. It's actually labeled here, and you can see some pigmentation in there. There's RPEs, which is fur back wrapped around the retina. So the individual color dot, you can see the cloud, as you can see, is across the whole tissues. But you can see the much less density in the places where you don't have nuclei, which are the plaques layer where the synapse forming. So there's less RNA there. So most of them do concentrate at the nuclei layer, which makes sense. So then we start looking at since we devised about 300 a panel of 368 genes that basically try to capture the subtypes of the mouse retina, and we start looking at some known markers, their level. And here's zoom into the outer nuclear layer where the photoreceptor cell reside. And then the red dots basically are cone markers. You can see the cone markers are primarily at the very top of the outer nuclear layer. And then I just really hand draw this cell boundary. And also, sorry, you can see there's also bright lines outline the cell boundary. That's where the cell boundary marker. One thing you can notice is the cells are very packed in the retina. That's quite different from many tissues, for example, like brain. So therefore, it is actually one of the first challenges we met is be able to segment the cell properly, so you know which dots belong to which cell. So if you just look at this raw data, if you just hand draw, and then I can easily pick out where's the cone cells. You do see some cone markers, outside the cell body, but they're really rare, like very small number. Once you see inside cell, they typically have multiple dots. So, and then if we overlay that with the rod markers, you can see rod markers everywhere because the rest of cells are rod photoreceptor cells. Cone cells is much less than the rods. It's about 2-3% of the retina, where rods is about like 60-70% of retina. So like 20-fold differences. And also the location of the cones makes sense. They are at basically the second layer of the top of the photoreceptor layer. So their location is consistent to what we know previously from other antibody staining. Similarly, we start looking at bipolar cells, which are basically one type of interneurons, as I mentioned, that reside in the inner nuclear layer. As you can see, the pan-BC, like basically all the bipolar cells will express these markers. You can see the yellow dots at the top half of the inner nuclear layer. So if we zoom in, and then we start overlay with different subtype, different subclass of bipolar cells. For example, on-bipolar and off-bipolar, both are cone bipolar cells and the rod bipolar cells. And then you can see them are not, unlike the yellow dots, they form a particular pattern, they concentrate on individual cells. So based on visual inspection, it seems the marker we know of seems like have a very nice pattern and then relatively low background. And then if you just step back to see overall how it's performed. So this is the two replicate. Those are two biological replicate, two different mice, and then are processed at different time. I use the morphage, the same probe set. As you can see, if you just like without doing segmentation, you just gather the total readout from those probes, and then each dot is one transcript basically. And you can see they form very nice correlation with R-square of 0.93. So this is really impressive, meaning it's very reproducible, even at the level which are really low. So we then I'll compare, okay, if you're self-replicatable, but whether it's same or similar to the RNA-seq. So we compare the morphage count to the bulk RNA-seq on the mouse retina. Of course, given that there are different chemistry and then the technology, but overall you can see a very consistent readout there. This actually R number is 0.8. It's actually better than if you just take the single cell RNA-seq data and then compare to the bulk, and then actually typically perform worse than that, particularly at the low end of the spectrum. So as I mentioned, segmentation, the retina is extremely packed, the tissue. So we find initially when without using the cell boundary staining, it's really hard to distinguish one cell from the other, but just based on the distribution of the transcript, they all form continuous cloud. Right? So therefore we find it's necessary to actually highlight the cell boundary use the antibody staining. So this is what it looks like, very nice staining. You can see you can easily from our eye easily pick out which cell belong to which. So and then we tried a variety of software out there like CellPulse and then Mesmer and other things, and then we're able to reach a reasonable segmentation. I still think there's room to improve, but at least if you just eyeball it, it seems quite consistent with what you will highlight yourself. So we feel our segmentation is reasonable, and then as I said, there's additional room to improve. So after segmentation, then what we can do is then we can assign individual transcript to the cell. And with that segmentation, as you can see, this is just the histology of the retina. You can see the outer nuclear layer, inner, and the RBC layer, right? And then you have basically empty space in the middle for the plaques with some cells, occasionally some cells. And you can see here, this is the entire section of the entire retina, one of the sections. And then the blue ones are the transcript that we're able then to assign to individual cells that is segmented, right? And then the red ones are transcript or dots, basically are not assigned to a cell. So you can clearly see the blue ones are basically corresponding nicely to the nuclear layers. You have this outer nuclear layer, inner, and then RGC, where in between, we essentially do not segment cells. It's very few cells there, and then you can see this as transcript. You can consider as a sort of background transcript that hold up there. So in general, we have like about 80% each of transcript is assigned to a cell. That is quite good. And then if you look at the inside of here, which there's no tissue, this very little signal, suggesting that the background signal is actually quite low. So also with the assignment of each transcript to the segment cells, then we can start to basically mapping the cells and then do cluster analysis, just as you have done with the 10x single cell RNA-seq, and identify the major cell types there. So here's the UMAP for Murfish. It is lower resolution for sure compared to the total RNA-seq. Nevertheless, it's clearly we can have a very distinct clusters, and this is the known marker genes, a few known marker genes that including the panel, their gene expression. As you can see, they're quite distinct. And then based on the marker, we are able to basically annotate each clusters. And then we are glad to see even for the rare cell type like horizontal cells and the RDCs, as I mentioned, only 1 or 2% of the retina, we're able to clearly identify enough of those. So to further investigate whether this makes sense, this is just a heat map for various of cell type specific marker genes on the cluster. And for example, RNL is a rod photoreceptor markers. You can see it's highly expressed in these clusters, which we basically know as rod, and so on and so forth. So we feel pretty confident at this level, the major cell types can be readily identified by the Murfish. So this is a quick look at the raw data to think about, why is that happening. So you can clearly see, for mural glial cell and rod cones, which are relatively simple cell types as mentioned, and you can see the markers. So let me orient you a little bit. So each column is one of the markers. Right, marker we in the panel, and each row is a different cell. We basically assemble to make it more visible, and then cluster them. So as you can see, the cell like round cones are very similar. They are all photoreceptor cells, and they are grouped together. And as expected, the interneurons are grouped together. And if you look at the markers more closely, the rod markers show very strong expression. Primarily only in the rods. If you look at column, you see they share some of them are photoreceptor cells, but there's a bunch of the rod specific ones, very little signal from other cell types. Similarly for the cones and mural glial cells. And then when you come to a more complex cell type like RGC and emigrant cells, you can see the RGC markers, which are sort of pan-RGC markers we have field put in there, show very specific RDC patterns where we're pretty much missing in other cell types. The same thing for emigrant cells and then for bipolar cells. On the other hand, if you start looking at subtypes, which you can see in the RDCs, we have a bunch of new markers there that really try to capture different subtypes, and they are quite sparse. That's expected because they are really rare. The cell type are really rare. And then the information there is probably limited in essence, right? This distinguish different subtypes. You only have a few markers trying to differentiate it. And then those markers are typically, when you go to the subtype level, they may not be so unique. It's unique or relatively unique in RDC, but it may actually express in other cell types. So that actually is one of the challenges you can see is given the limited size of the the panel, if you want to find, to go to the subtype level to get all 100 of them, particularly for rare ones, it is challenging because meaning you need to expand the markers. That's what we think for those cell types, be able to distinguish them. So, and then after checking the data, and we feel pretty confident, then we can start assign them back to the section and see where they're at. As I mentioned, this is the same retina I showed you, and this is zoom in, and then you can see the outer nuclear layers here, and then the pink are rod cells, and then green are cone cells. As I mentioned, the cone is on the top of the outer nuclear layer here, and then you can see also there's some the pattern that in the inner nuclear layer where the top layer is mostly bipolar cells and the amiculum cell more at the bottom half of the inner nuclear layer, where RDC, which is the brown, is in the RDC layer. You can see some blue actually displaced or like amiculum cells in that one. I will come to that a little later. So overall, the structure for the main cell type, the spatial pattern is quite consistent what we know. We're pretty happy about that. So now we want, of course, we don't want to stop there. We don't learn necessarily too much if we only stay at the major cell type level. So we want to get to the subtype level and then to see how much we can push the technology. So this is so we start, basically, we take all the bipolar cells and then we start doing high resolution, increase the resolution to the subtypes. So here's the Murphy's data along. You can see, in fact, we can identify pretty much all 15, there's 15 different bipolar cell types. We can identify all 15 of them. But you can see like RBCs, right? As I mentioned, they're rod bipolar cells, and then BC1A through like 4 are basically off bipolar cells, and there's also on bipolar cells. So anyway, but you can see there's a lot of sort of touch each other, right? The cluster they are, because as I said, those subtype level they are driven by just a few markers. So and it's sort of noisy. And here we just go to a slightly lower resolution, so we can group them into the on and off and the RBC. They're sort of separate. But once you go to either the further level, then they become a little bit hard to say some of them the assignment. So how to deal with that? So one thing we're thinking about is that can we then leverage the RNA-seq data, single cell RNA-seq data to help resolve those subplots. So that's what we did. We basically run a co-embedding by combining the single cell RNA-seq with the Murphy's data. And then as you can see, instead of this sort of semi-separate clusters, and they can be separated much better into very distinct clusters, and primarily driven by RNA-seq resolution. So as a result, we can get all 15 ones. And with this information, then what we can do is then we can explore whether there's any spatial patterns that was not obvious before, and without this sort of data. So the first thing we check is whether the number of subtypes, the population, is actually consistent with what's known from RNA-seq. So here's the proportion, and then this Murphy's based on annotation is Josh Chen's single cell RNA-seq on bipolar. As you can see, they are the proportion of our bipolar cells is quite consistent from the RNA-seq, which is slightly lower the rod bipolar cells, where the others are quite similar. So we not sure exactly why the difference is there, and I think partially because of the data, but partially could be just slightly different mouse or technology used. But overall, the trend is quite similar. So then we can replace those individual subtypes into the retina. You can see they form they occupy the upper half of the inner nuclear layer. So then we use the Giotto to start explore any sort of spatial relationship among them. So this is the output from the Giotto, and they basically show you once you have the edge, meaning they are more close together than by chance, and then if you, so you can see here like BC1A, 1B, which are very transcriptally very close, seems to pair more often than by chance. So that's actually reflect here, a quick section here. 1B is in red and green is 1A. I can see they often form like a dimer or together, or maybe heteromer, meaning one red and green. And then if you can go out a little bit, you can see, have like 1A, 1B are more closely related with 5C and A than 5B. And then if you look at that, they seems occupy a particular maybe sublayer of that. And this data is a little bit, sparse, but we have that. It seems like they occupy maybe a sublayer more often than others. So suggesting there's some interesting maybe cure for them during development. And of course, overall are quite random. Most of the bipolar cells are placed randomly, so it's not like a very unique repeat cassette there. Now, one thing interesting is the spatial close proximity not necessarily correlated with the on off. So you can see, for example, here, 8 is in this branch, and then in the 1A, 1B is in the other branch, and then they actually are more closer than the cells that are within the same branch. So the spatial placement is not necessarily correlated with the transcriptome closeness. So we want to further push to see, okay, now bipolar cells is complex, but it's not as complex as, say, endocrine cells, RGCs. So how can we do good job in the endocrine cells? So here's the co-embedding results. So with Murphy alone, you can probably get around like 15 to 20 endocrine cell clusters. If you leverage on RNA-seq, you actually can get about 40 to 50, almost get all of them, identified. So then we can again look at their spatial relationship. One thing is about mostly are sort of random. There's maybe 2 clusters there. So one of the cluster called our eyes that it seems like map very close to RDC, which is supposed to be a different layer. So this is how it looks. There's 5 endocrine cell clusters that suggest to be mapped spatially close to RDC layer. And then if we when we plot it on there, as you can see, yeah, some of them are instead of mapped at the internuclear layer where they're supposed to be, they are in the RDC layer, which is known for some of them. For example, AC17 is called starburst endocrine cells. It's very distinct endocrine cell type. Has been known some of them, a significant portion of them somehow mapped at localized at the RDC layer, what we call displaced endocrine cells. But it's also 4 other AC types that are identified in our study. It's also mislocalized to or displaced to the RDC layer. And interestingly, all of them are GABAergic endocrine cells in cholinergic. So we're in the process of validating the finding by other RNA-scope probes to basically directly do in situ hybridization to make sure that what we see here is also can be reproduced by using a different method. So this is just a zoom in. You can see there's plenty of endocrine cell 17, which is basically a starburst endocrine cells displaced, but also with others. So when we think, we're wondering, like, since we see that is the number of cells really rare, so we're wondering whether that's just because of misassignment of the cell types or some background. And then what we start looking at is the remaining endocrine cell types and their distribution. So just a quick zoom in the blue representing endocrine cells we detect have displacement pattern, and you can see there's a lot of blue dots in the RDC layer. And the red are others remaining endocrine cell types that we don't see a displacement. You can see pretty much all of them are indeed in the nuclear layer. Only a few dots that potentially are synchronized. Maybe they are sort of background. So overall, we don't think it's assignment or background. It's probably the real biology. So finally, just the RDC types I showed you here. The RDC is very complex in the mouse. And instead of 15-ish in human, there's probably 40 to 50 different types in the mouse. And as you can see, even they are rare and very complex, we will be able to separate out and identify all of them, or most of them, in our data set. So that's the spatial map of RDC. You can see beautiful alignment, and then we start looking at their pattern with in the middle of doing that, and we don't see a very unique spatial pattern on that. Seems like more or less they are sort of random. So given the results, we feel like overall the technology is quite robust. Then we are thinking about, okay, whether it's possible. I mean, it's great to have a spatial reference map, but then you really want to build on that reference map to start looking at disease and mutation, things like that. That this technology sufficient to do that? So one thing we would like to try is take a mutant retina and see that known to affect a small subset of cells, and to see whether we can detect that with this technology. So one of the mouse we test out is the LHX3 mouse, which has been known to express specifically in bipolar cell types and in subset of bipolar cells. And then this is a transcription factor, LIMH-X3 homodomain 3, and you can see in the wild type, and we include that transcription factors in our probe set. So you can see as consistent to previous known that LHX3 is primarily expressed in the nuclear layer where the bipolar cell reside, especially the top half. And then in the knockout, it's a 6-3 create like conditional knockout, and then we can see the blue dots essentially are gone. There's a few dots there, probably partially just due to it's not complete knockout, it's a conditional knockout. So maybe 95% are gone, but there's a few left. So and then we start looking at overall what it looks like between the knockout versus the wild type. As you can see, the only gene that reduced in expression detected is LHX3 compared to wild type, which is really assuring. And then we start looking at more closely on bipolar cells. Then what happened to bipolar cells? Here's the wild type reference map. Here's the LHX3 conditional knockout mouse. So you can see almost all the clusters are there. The only two things are missing is BC6 and BC1B. And here's the quantification. You can see the dramatic reduction of number of BC6 cells and the BC1B in the knockout mouse. So this is not the case for the rest of the cell types, for example, amicron cells or RDCs. There's no, for all the clusters we look at, there's no statistically significant drop or increase in other cell types, which is also consistent with the role of LHX3 specifically in bipolar cell types. So just to summarize what I have told you today. So we feel the morphage is highly robust and reliable in detecting transcript in the retina tissue. And we think they're probably true for other cell types, tissue types, which just a lot of people already published. And I think vast majority of cell types can be identified by morphage, but probably either you need to increase the number of markers you use, specific focus on that cell types, or you can in conjunction with the RNA-seq to achieve that. So we feel that we do start learning some interesting insight of biology because the single cell spatial data once we have it available. And I think this morphage is not only robust, but very sensitive to detecting very subtle changes in unique cell types in our data. So for us, we're currently working on combining the single cell RNA and the TAC-seq with morphage to produce this atlas map for the humans. So right now, what I show you is the mouse, and right now we're already designed the corresponding probe set for the human, both for the retina, also for the anterior segment. And then we want to basically build a spatial map for the human eye as part of the HCA project. We also would like to apply the morphage to look at mouse and human anterior segment, posterior segment, and also we're already testing the brain, and it works really well. And in fact, work on the brain is easier, less challenging than the retina because the cells are more sparse. And finally, we would like to apply the Murphy's to looking at development process and then the mutant and then disease tissues. So we can learn something local change, for example, around the lesion side, which is probably missing by current single cell RNA-seq because the size is very small and then it's hard to distinguish cells next to the lesion versus cell far away. So this will be critical usage. So with that, I would like to thank the people who produced the data I showed today. So this is really primarily produced by Johnny, a graduate student in the lab, and also by Selma. And then the analysis is done by Johnny, collaborate with Li Jin, with a postdoc in the lab, and then Qinai is a senior student who actually started the project. So we are very close. We have close collaboration with folks at the Jin and George and John. And then this is not possible without collaboration with a bunch of people like Jeff, as I mentioned, who is the co-inventor of the method, and also Jason, Jinzhuang, and Ken for analysis, and Lingan is the one who provided us the mouse, mutant mouse LHX3 I showed you today. So we are funded by NIH and the Retina Research Foundation, and also Chang-Jasberg Initiative. Thank you very much for your attention, and happy to take questions.
Thank you, Ray. So we are now open for questions. You can either raise your hand or type in or unmute yourself to start asking questions. For raising the hand, just go to the reaction button. David Ginsburg, go ahead. David, you need to unmute yourself.
Sorry, that was really fascinating. Thank you. One quick question. I'm particularly interested in endothelial cells, and I noticed that in your later subsets, you didn't seem to be resolving them. Can you tell us a little bit more about what you saw in the vasculature?
Yeah, so when we do the RNA-seq, single cell RNA-seq, endothelial cell there's very small amount in the retina, like less than 1%, I would say. So when we design the probe, actually we, because the way we do section, right, the endothelial cell is just like a small hole there. So we didn't put any probe that target endothelial cells in our current probe design. We're actually doing the second design that. But also another thing, you really want to visualize them, we probably need to use instead of cross section, we probably need to do whole mount, just different ways, so you can see the vasculature, which also pose a lot of technical challenge in there because the thickness of the tissue and then the just optic. So yeah, that's something interesting, but I think it's quite challenging at this point. Yeah.
Thanks.
Jillian, you can go next.
Great, thank you very much. That was really nice, Chen. Dr. Chen, I wanted to follow up on that question about whole mounts, because I think spatial information for a lot of these really sparse amacrine cells and ganglion cells would be very interesting on the whole mount retina. I was wondering if you guys, because obviously if you guys had any thoughts about how to begin to resolve that, maybe even just the ganglion cell layer or something like that.
Yeah, that's definitely a great interest of us, right? Because you want to see the dendritic field as well. I mean, if you not only the cell body. So we actually tried once by doing the whole mount, and then the signal quite fuzzy because the optical. So we are thinking about two ways. One is just do a series section, so we can just really reconstruct that. So that's one approach. The second one is instead of just take the full mount, we put a whole mount, and then we need to do some sort of section. We can butterfly it and section it, and then it is possible to do like around 15 to 20 micron. But it's hard to do more than that. So currently, just because the light scatter is just the laser, it's just you don't get a good signal and hard to resolve. So yeah, it's definitely something. And also, I know I talked to Jeff. I know people also doing the series, just like the prior EM type things. They're taking one the series section and then process it, and then to the second one, the third one, the sort of flat mount sections. But that's just, you need to have a lot of section to go through the stuff, and then and also pretty intense in terms of the reconstruct that 3D map, right? It's not, it can be done, but we just haven't tried it
We'll go to Ahmed, followed by Sue.
Yeah, hi, can you hear me?
Yeah.
Yeah, so really great work. Thank you so much for the presentation. So I have a quick question regarding how do you integrate these single cell data with the spatial data to learn like would have better clusters for the cells coming from the spatial data? Like which features do you use the overlapping features only, or like which method for integration of the data?
You mean the co-embedding of the single cell versus the Murphy data? Is that what you mean?
Yeah, how do you map the Murphy's data to?
Yeah, so we tried a different thing and different software. So currently what we do is we just take the, the variable genes in the RNA-seq and then take all the probe set in our panel, like 368 of them. So we find actually if you just restrict to say only 368 from both, it doesn't really work well. And then if we only restrict to a subset of, like say, bipolar cell types, it doesn't work well because the other marker are not intended to be distinct bipolar cell. It still offers some information there. So it's actually making it simple. You take the whole RNA-seq data and take the whole probe set data, and then the question is how you co-embed them, right? So the idea is like since they're the same cell types distribution, so they should have in some low dimension space, they should align them each other nicely, and then you should be able to do that. So we tried BindSC, which we and then Ken Chance, our collaborator co-developed, and then called BindSC. You can find that already in the GitHub, and then it works pretty well. You do need to tune a little bit because here the resolution depends on which level. If you want to go to the really high resolution as we're trying to do, you need to rely on more on RNA-seq, so you can put more weight on RNA-seq instead of the Murphy, right? And then you can try to sort of line the Murphy on top of it. So that works well. And then we also tried other things like SCVI and others. I think so far BindSC works really well. And then and also we recently tried Tangram, which it works, but you need to be really careful because it's borrow some things like from outside. You need to sort of do some tuning, and they still can get pretty good. So happy to talk to you more about how we achieve what we have so far. But with that said, it's very clear that none of the methods we tried so far are perfect. So there's a lot of misassignment. And then one of the difficulty we have here is like we don't know the ground truth per se, right? We don't know which cell exactly what is what. So but we basically develop a set of metrics to evaluate different co-embedding method, and then to see which one works well. But from our metric, I know none of them are perfect, particularly at the very high resolution. So I think there's room to improve for sure from software.
Thank you.
So you can go ahead.
Great talk, Chen. So I have a few questions, and some of them may be a little bit technical. I'm just curious to see how to benchmark potentially Murphy's data. So what % of the genes, so you have around 500 genes, is that correct, that you tile? What % of those genes are actually detected in your experiment?
Yeah, so go ahead. Yeah.
Go ahead, you could answer and then I'll ask the next one.
Yeah, I saw you finished. Sorry. So yeah, the number of genes we try to detect is 368. So those are the marker genes from the RNA-seq. We pretty much detect all of them, maybe one or two drop off, but all of them we can.
That's pretty amazing. So you could cluster based on a few number of markers. You could get that nice clustering of all the BC cells just by with so many different subtypes by the 300. That's pretty impressive.
Yeah. Because we choose them, right? Choose the most variable gene based on the RNA-seq. So those are, we choose the markers. And right now, our second version is there's this server software available right now. You can feed them the RNA-seq data and then let it spill out a set of genes, like typically around 500, to capture the topology of all the RNA-seq, cloud or UMAP. When we look at that one compared to our handpick, the top markers, there's a lot of overlap, but there are some additional genes that seems the software think is useful. So we haven't tested to see whether it will improve the resolution.
So with all these single molecule approaches, there's a lot of puncta that are formed, but actually what % of the puncta are actually decodable in your hands in your experiment? So how many puncta are just like random autofluorescence that you're detecting. And I guess I don't know how crude you get the data from Visiogen. So I'm not sure. It depends on what level of filter they give you to be able to identify this.
Yeah, so this, the Murphy have a built-in of error correction. I mean, not error correction, error correction. So it's possible doing any hybridization step, you're supposed to have a 1 and you say 0 or vice versa, right? So I think based on their paper, it says the miss detection is about 3%, right? Overall, like 3% you will misassign them. So that's what we sort of see there. So and then and they have particular each probe we design is pretty sparse. There's many cycles. In our case, like I don't know, it's 20-some cycles hybridization or something like that. So there's a lot of error, robust built in. So if the code doesn't match with what your codebook will get rejected out already. So I do not remember what % of the probe reject because of that. I can check out.
Not necessarily. I think just calling any real, like anything that's diffraction limited that becomes a real puncta, of that, what % I was curious that actually codes a gene versus a regular just background. But I think I could talk to you more offline about this.
So we know, like once they passing their pipeline, they say this can be assigned a transcript, then about 80% can be assigned to our segment genes. Yeah, the segment cells. Yeah. So
let me go to the question chat and come back to Kyung-Yang and David. Josh was asking in chat, have you used location information of the data that might be missing from the single cell RNA-seq data?
Yeah, so that's what we have been using. Like once you assign, you down cell segmentation, assign transcript to it, then you can start looking at multiple levels. You can look at the cell relationship, right? Cell type relationship. We do see, so the conventional idea, as we go in is like there are random distributed, right, within the layer, right? Different subtypes. They don't seem to be, but what we see, for example, for bipolar, there are some close propensity between something 1A and 1B. They are really close. They like to form heterodimer, like next to each other. They want to be neighbors. But I will say there's tools available to explore those relationships actually very limited. There's a few tools there. I think again, there's a lot of need to do that. Another level we haven't done, we're in the process doing is you can look at the gene expression spatial level. For example, it's very common during development, you have a gradient, right? So if you have an organizer and then you have a gradient of BMPs or things like that. So that actually when you need to, because most of the genes are not including our Murphy probe set. So that's why we need to, during the co-embedding process, also you allow you to do imputation. So you can sort of impute the entire transcriptome instead of testing it. And then with that layout there, you can look at gene level, the gradient, or local things, and there's software developed. And there's also some additional things we are just about to test it out. And I'm pretty sure we'll find something there too. So at 2 level, you can use the spatial information, at least from my perspective.
Hey, can I just follow up real quick? I was curious specifically about single molecule information so that you can actually look at like subcellular localization, nuclear versus cytoplasmic, things like that.
Yeah, it is possible. So it's a little hard for the retina tissue right at this point, because what I show you is the most of transcript really clouding around the nuclei, and they have long processes, right? It's hard to know when they go to the plex layer, and it's really hard to know which process is from which cell. But one thing we are usually, for example, what transcript are enriched in the synapses, but not in the cell body, for example, right? So then you need to either have the probe, you have candidate, so you can probing it. That's one thing, or you need to revert to actually sequencing-based method, in my opinion, to be able to sort of get, a candidate, or then you can validate that. So for long, the cell with long processes, be able to stand and then call out the whole cell body boundary, that's actually more challenging. Right? They travel so long distance, you need to have, they're not going XY plane, they also go Z plane. So, to find that actually is definitely a big challenge. But the resolution is there. You can see that, if you have the right cell, entire cell call out, you'll be able to look at subretinal, subcellular like cytoplasm or nuclear. Then you can see it. Yeah, but that is that.
Yeah. Let's go to Kyung Yang, and then David had the last one.
Yes, I also have a kind of similar question about the resolution. So what is the limit of the resolution limit of your or analysis, some pixel resolution level? And also my question is so kind of mostly focused analysis on the a cell body, but is there any way of the show some extracellular matrix or some plexiform layer to show some interaction between the neuron or other cell? Is it possible?
Yeah, so I think in the first one, the resolution. So I didn't pay too much about resolution, but what I can, based on my conversation with Jeff right now, is that it has a resolution to pick out one transcript, right? So if it's very accurate in that sense, it's like at the sub-micron level. So and then if you have say 20 transcript, you should be able to see all 30 transcript in the or 20 transcript. You can see all 20 spots in the cell. So it's quite high in terms of, for us, like our cells at least 10 micron level, and that's definitely way higher than needed at level. And then in terms of the interaction, yeah, it's definitely interesting thing. So I think the difficulty I see for the retina is because they send out these long processes, right? So it's hard to tracking them, as I mentioned. One thing, of course, as neuroscience is that interesting how they talk, right? The cells next to each other doesn't mean they talk to each other. So I think this spatial coupled with the tracer, right, viral tracing technology will allow you to actually see which one talk to each other in space. Another thing I think is useful, the spatial one, is that when you have a single cell RNA-seq, one of the analysis people a lot of time do is ligand receptor interaction, and then you can get a lot of false positives. If the cell are not next to each other, if cell surface, receptor will not, talk to each other at all, right? Because they're so far away. So I think with the spatial data, actually can very efficiently filter out those false positives. So that's be one of the potential usage for study cell-cell interaction.
Thank you. David, thanks for your patience.
Hi, thank you for a very interesting seminar. I was just curious what marker you were using to distinguish boundaries between cells, because this is always an interesting problem in the retina in terms of assigning in situ signals.
Yeah, so in fact, the membrane surface markers come directly from Gen, so they have 3 different ones. And then we ask them, they don't tell us exactly which one. So there's 3 different ones and then different channel. In the retina, I only can tell the channel 2 works really well, but 1-3 doesn't work really well. But it's not always, because in the RGC layer, channel 2 works really well for most of the layer except RGC, which are very heavy, high dense membrane. And then the channel 3 actually works well. So we need to mismatch different things there. So they don't tell me what exactly surface marker they use. My understanding is some surface antibody they conjugate.
Okay, thank you.
Right, we have one more question from the chat. Sesu was asking about segmentation. Are you assigning transcripts to nuclei or cell body, and how do you benchmark errors for segmentation?
Yeah, that's a great question. How do you get segmentation right? So we have a multiple layer. One is you can, after segment, you can visualize it, because our eye and brain are actually quite good at segmenting. If the staining is good, you can see it. So that's one. And the second one is we have some sort of summary statistics about it. For example, the size of the cell body, the volume, right? So if you segment wrong, then you're going to have a lot of outliers. And then the location of the cell body, where in the text layer there's very few cells. And then the third thing we try to do is, like, if you segment correctly, then you assign the cells, or the transcript should be accurate. So then we also look at that level at transcript or marker level to see whether our segmentation makes sense. Because, for example, you shouldn't have cells that express both bipolar and ambivalent cells together, right? If there's a bit there. and very few should be like that. So those are the ways we use. We cannot be 100% sure the sampling is accurate. In fact, it's a definitely active research area people can improve on. And there's several software there. We basically use like 3 or 4 summary statistics to try to benchmark which one works well. And so far, we're doing the cell pulse is the one works well for us.
Great, thanks, Ray. So we're at the top of the hour. Don't forget to thank Vanessa Evans, who is the managing director, helped to organize this event, and Sunisa Nagras, who is also the faculty lead for these monthly seminars. To thank all of you for attending, and Ray, thank you greatly, for joining us from Houston.
Thanks, everyone.
Bye-bye.